It is 2:02 right now, so maybe we'll just get started here. So thank you everyone for joining our webinar on how to master designing protocol figures. So here's the agenda for today. After a brief introduction, we're going to dive into the best practices for building a protocol figure. We'll then spend a little bit of time outlining the top 5 tips for ensuring that your protocol figure is as impactful as possible. At the very end, we're going to be doing a live figure makeover to tie everything together that we've learned. And this is always a crowd favorite, so please make sure to stick around for that. We also have some information at the very end about institutional pricing and different links and resources for BioRender, so some valuable information at the end there. Okay, and with that, we're just going to give a quick introduction to ourselves. After the introduction, we'll probably turn our videos off just to kind of focus on the presentation, but just wanted to introduce ourselves. So my name is Mina. I am one of the science designers here at BioRender. I have my background in biology and biochemistry, as well as a master's in neuroscience and a PhD in medical science from McMaster University, which is in Ontario, Canada, for anyone who's international. And I'll pass it over to my colleague, Sally.
Yeah, hi everyone. My name is Sally. I'm also one of the science designers here at BioRender. Like Mina, I also did my schooling in Ontario, in Canada. So I did my bachelor's in University of Toronto and then transitioned over to do my PhD studies also at University of Toronto, focusing on physiology.
Awesome, thank you, Sally. Okay, so before we dive in, we always like to share our mission here at BioRender, which is to accelerate the world's ability to learn, discover, and communicate science. And to that end, we know obviously that a lot of scientists have to communicate about their protocols. It's an essential part of the science communication process. And accurately depicting protocols is really essential for effective communication as well as experiment reproducibility, as I'm sure we're all aware. So as we get started here, we're going to launch a second poll. So maybe we'll close the first poll here, some really interesting results. And maybe we'll go ahead and launch the second poll. We're really interested to know what everyone here struggles with the most when creating a protocol figure. So feel free to start answering that poll. Oh, already some really interesting results.
Yeah, well, let's some of our answers roll in to see where we're at. It's
a really interesting kind of even split across the board here between getting started, choosing the best layout, finding graphics, and finding protocol figure making a little bit time consuming. And these are definitely all pain points that I'm sure Sally and I in our previous academic life can really relate to.
Mm-hmm. Yeah, for sure.
So do we have a winner right now? It looks like maybe choosing the best layout is just edging out finding the right graphics and having it be time consuming. Really interesting. Okay. So feel free to keep answering that as we continue here. So I guess the first question that we want to ask ourselves is how we can approach making a really impactful protocol figure in BioRender. So I want you all to imagine that you're planning a trip from San Francisco to LA, and you need a reference map to know exactly how to make that journey. Okay. Next slide, please. So we have a couple of options here for maps that we can use. And the one on the left is a really beautiful map. It has a lot of really interesting illustrations. It's very colorful. It looks really, really nice. But if you compare it to the Google map image on the right, you can see that the Google map image is a lot easier to follow. It has a much easier flow and composition. It minimizes the use of color and really uses color strategically to draw attention to key elements that are needed for that trip. So it has a very clear starting point at A, a very clear ending point at B, and a very clear path in between highlighted by the blue. So it really takes advantage of those design features to tell the story a little bit more effectively. So we want to try to keep this analogy in mind as we really dive into how to master making an amazing protocol figure. And with that, I'll pass it over to Sally.
Yeah, cool. Thanks for that introduction, Mina. So let me also turn off my video just so that you can focus on the screen here. Yeah, so hopefully with that introduction, everyone now understands exactly why it's important to make clear, concise protocol figures. Now we want to move on to the part of the webinar where we give you the tools to help you actually the next time you will set about making a protocol figure, starting with steps on actually how to build a protocol figure. So let's get right into it. So as science designers, Mina and I make a lot of protocol figures, and over time, we have realized that following these 5 steps that we have outlined for you in this timeline really helps to make our figure making streamlined and as easy as possible. So I think some of the comments here was that it takes a lot of time to make figures, so hopefully this helps. So here are the steps that we recommend you try out the next time you go about making protocol figures. But let me also, of course, go over the steps in more detail so you know what each step is about. Cool. The first step before you even open BioRender is to consider the context and the audience that your figure will have. This is important because the amount of information you will have to include on your figure will depend a lot on what the figure is for and who the figure is for. So you could imagine, if you are making a figure to present to the general public, a lot more information would have to be included on there to provide them the proper context compared to a figure that you're going to present to your teammates. So yeah, make sure you thought about both the context and audience, and you're sort of in the right headspace before you truly start making your protocol figure. The next step is to make a reference board. So what do I mean by that? I mean to collect all the visual and written materials that you want to reference while you're making your figure and add it all to a blank BioRender file. So let me demo this for you. So here is a blank BioRender file, all white, nothing added to it, that I have opened up. Let's just zoom out for a little bit to show you that on the top, I've gone ahead and added a timeline of steps that I was referencing earlier so that you can follow along as I demo the figure making for you. So let's imagine a scenario where I have to create a protocol figure about Western blotting for my teammates. So I don't have to go into super detail and it could be quite simple. So to build a reference board, I will just simply onto this file, start adding in images and text that I want to reference as I am making the figure. So I'm just copying and pasting from other tabs I have open at the moment, various images that maybe you have saved on your personal desktops or things you found during Google searches that you want to aspire your final protocol figure to be, or even maybe you have some text from your Word docs that outline all the protocol steps you have that you want to include in the figure itself. Feel free to go ahead and add that in as well, so that you all have it in one place. And yeah, and you want to keep doing that essentially until you get to some place where you have all the references added on. So something that looks like this. And we really recommend this step because before I even knew about BioRender, when I was a grad student, really I didn't do this step, and it made it really frustrating because I had all these references everywhere that I had to gather, and it was super frustrating to find the information I wanted to find. So adding this step in really helped streamline my figure making process because it was super easy to find all the information I needed. So really recommend adding this step to your figure making. Okay, so the next step after creating a reference board is to sketch it out. So this is the step when you consider all the references that you have collected on your reference board and sort of start brainstorming what your figure is going to look like, and then draw out the layout you kind of want to achieve. So you could do this on BioRender. I like to do that just because the reference board is already on BioRender, so why not just kind of continue the BioRender fun? Or you could do this, physically with a real paper and pencil. Sometimes that helps with the brainstorming process, but for the purpose of the demo, I will just do it on this board. So when you're setting about to do that, you could do one of two things. If you already have premium access to BioRender, you will have access to our slides feature. So that's actually how we've been doing this presentation within BioRender itself. If you go to this side over here and show slides, you'll see our little slide deck. So if you have this feature already for yourself, what you can do is just add a blank slide and then just do your sketch over here, and then kind of go back and forth between your reference board and your blank file to make the sketch. I actually like to do it on the same slide. What I like to do is just select everything and then push it over to the side so that I can still access the information just by scrolling to the side like this. And I still have an empty canvas that I can add things onto. So what I will do to sketch it out is kind of look at my references here and sort of decide kind of what I want my layout to look like. And upon pondering, let's imagine that I mostly want to make my figure look like this reference over here. So to sketch it out, what I will do is I'll go back to my canvas and sort of block out the panels much like it is here. So to do that, I'll go to the top toolbar and click the shape option and just pull out a simple rectangular shape, drag it out to block out the first step. And then from then on, just simply copy and paste to make panel number 2, panel number 3, panel number 4, so that I can get somewhere close to this. Cool. So I think I like what that's looking like, and sort of to help the future audience of this figure follow the steps, I think I'll add numbered steps too. So you could also easily do that by going to the shapes, going to the text shapes and pulling out this option here. This is one of my favorites because, you know, it's just super easy to pull out a clean looking numbered step without having to, you know, fuss about with the formatting. So there you go. Of course, this is just a sketching option, so I'm just adding in the ones, but later as I refine it, you can change it to a 2 or a 3 or a 4. Cool. At this step, I'll also pull out all the icons and shapes that I think I'll need. So I'll basically go to this icon library section over here, and then this is going to be about a Western block. So just type in Western block and see what comes up. And I will literally just pull out all the icons I think I'll need at this stage. So I see some gel running machinery over here. Just pull all of them out. I'm not fussy at this stage. I just literally pull out what I think I'm going to need, and then I'll make the decision later. Based on my reference, now that I can see it, this will be about the gel transfer. So I'll also, you know, pull out some gels. And yeah, you want to just at this portion, just pull out all the icons and shapes you're going to need until you get to a place where all the major portions of your sketch are laid out like this. Cool. So now that we're at, we have completed step 3 and we have all sketched it out. Now we want to move on to the step 4, which is the arranging icons and shapes portion. So this is the step where you want to narrow down the actual icons and the shapes you're going to use. So this means, you know, access icons that you pulled out just when you were brainstorming, you want to sort of delete. And the rest of the things you want to place more mindfully and purposefully. So for the demo, let me just focus on this first panel over here. So, I mean, I don't need 2 gel running machines, right, to show step 1. So I can delete that one. And then this is sort of now over to this side, which looks odd. So maybe I just pull it to the center over here. And then now that I've seen this sort of placeholder box that's blue, I think that might be distracting the color. So what you could do is click on it, open up the side panel here where you can see all this style options, and then just click one. There we go. I like the white one with the plain black outline. And now that I see it, I think I could make this less tall as well. There we go. Oh, and also this is a step you will want to start adding text. So you can add any needed subtitles, things like that, and adjust the placement. There we go. And you want to do this all throughout your figure until you land on a draft that you're pretty happy with. So something that looks like this, where you have all the proper icons and the excess deleted, essentially, with the text written in like that. So from this point on, then the only thing you have to do is do step 5, which is to fine tune. So this is a stage where you start refining your figure a little bit further. So just look at your figure, do any last minute touches you want to do. So this might include things like changing the color of icons, for example. So now that I look at this gel here, it's kind of purple, and that's not what it really looks like in real life. So maybe you just click into it, open the edit feature, and then select a color option that you prefer instead. Maybe you want to bold all the text just to make it even more stand out like that. Yeah, and you just go about making the changes as you want. Maybe even delete these boxes because there's a lot of boxes, a lot of rectangles here. Maybe that's distracting, so you can even delete that. Like that, until you land at a draft that you're really happy with. Something that looks like this. And that's all the steps. Cool. Are there any questions at this point? Maybe about the steps?
So Sally, we did have one interesting question. Maybe we'll quickly demonstrate live. I was just typing the answer out for it, but we had a question about copying and pasting in BioRender. Do you have to actually copy and paste, or is there a shortcut that you're using to replicate things over?
Yeah. So there's various things you could do. Yeah, for sure. You can click, yes, I think you mentioned Option click, I think for Mac, or Alt click for PC. So I am pressing that button now and then just clicked it and then dropped it down. Oh, sorry, I let it go too early, but you can do that. Or you can do select, the good old Control C, Control V. You could also do it copy paste like that too.
Great. Maybe we'll take another quick question. How do you change the canvas size?
Nice question. Okay, so how you change the canvas size, maybe you noticed as I was fine tuning, let me go to the previous, there we go. So if I wanted to change canvas size, what I would do is if you go to this top toolbar here, you'll notice this canvas size option. You click that and these little black marks will show up. You can basically click and drag. You could also do it numerically here as well, but I like to do this so that I know I can really control where it ends and where it starts like that. And then you just click apply.
Yeah, a lot of really great options. And you know what, maybe we'll take one more. I think we're actually okay on time. There's another interesting one here for Sally. So in BioRender, is there a way to edit grouped icons and have it affect all at the same time? Like an antibody fluorophore pair, can I change the color and can I have that affect all of those in the paired group?
Cool. So I think, let's see if I could find a group here and then open the side panel. So if the group is mostly just shapes like this, you can just go ahead and click the group and you'll see that the style option exists. And then you can just literally go through the options or even go to this area here where you can change a color to be literally what you want, anything you want. That's an option. If it's something like antibodies and shapes where you have a combination, oh, sorry, I have to click out of this. Then you might have to, so let me just group these so that I could demo it for you. So you can't do the color changes with one click, unfortunately. The same functions don't show up, but you can just enter the group and then apply the changes that way.
With the one caveat here really being, if you have a group of all the same icons, let's say you have a lot of dots or a lot of antibodies and it's all the same icon and they're all different colors at the moment and you just want to select all of them to make them the same color, those ones can be changed together. And we actually have an option if you go to edit and kind of scroll down, you can see that you can select all the same options,
Mhm.
all the same icons, so that everything gets selected that's on the canvas, that's the same icon, and you can do it that way.
Let me actually do this. So you can do this. You
Yep.
can select the same icon. And so you'll see that those antibodies were selected, and then you can just change it. You can see that the changes are applying to both.
Awesome. Yeah, great questions.
Cool. Then for the sake of time, maybe then we'll move on
Yeah.
to the next slide. Awesome.
Okay, yeah, that was a really great presentation, Sally. I think it really highlights some solid steps to just the figure making process in general and how to build a really solid protocol figure. So right now what we're going to do is we're going to dive into our top 5 tips for making sure that your protocol figure is really as effective as it can be. So these are our top 5 tips. And really what you want to do is ensure that you're following a really simple composition. That's pretty key. Ensure appropriate text hierarchy, group things together where possible. You want to ensure that ensure appropriate icons and text proportions. And then you want to simplify. So at the very end, we'll talk a little bit about how to refine things by just simplifying as much as possible. So I was really interested to see in one of our polls that one of the biggest pain points, I think just edging out, in first place right now is choosing the best layout. So that's really what we're going to talk about in this tip. So what you see on the screen right now are the main compositions or layouts that are most effective at organizing visual information. And why might that be? Well, it's because all of them read either from left to right, top to bottom, or a combination of the two. And that mimics how we read text. So next slide. And these can be used for different purposes, and they can actually be used in combination as well. So if you have a really complicated figure, you might use a combination of these. But for more simple figures, simpler protocols, these are kind of our takeaway points. For protocols with few steps, a simple unidirectional composition is the simplest way of organizing your figure. So just go left to right and make it really, really simple is going to be the easiest for your audience to follow. For cyclical processes, you want to make sure that the steps are reading clockwise, because that's how we tend to read the clock, with the starting point placed either in the top middle or top left of the circle. So in this case, in the example that we see here, the patient would be the starting point and kind of the top left position, because that's naturally where our eye is going to go to kind of start reading. For protocols with many steps, a Z-shaped composition is going to be the most effective way of organizing your figure. So this actually retains the left to right reading order for both rows of information that you're showing, while still being able to show multiple complex steps. And finally, we have an example here for protocols that have either a converging or diverging step. We like to use a simple fork-shaped composition that reads either left to right again or top to bottom. And we like to avoid diagonal lines. So, just having things more linearly going from left to right or top to bottom, even when there's a fork, is going to be a lot easier for the eye to follow. So here we have an example of a figure that we made over, where really the main problem with the original was the lack of clear direction of information flow. So we can see that just reconstructing the composition really allows the eye to trace out the intended pathway of information a lot easier. So you can see on the example on the left, your eye has to go up and down and in different directions and kind of trace backwards before you kind of get to the final position, but it's still not really clear that I followed the correct reading order. And all the same information is presented in the makeover on the right. It's just the composition is refined so that everything is a lot more linear. And we'll talk a little bit about some of the other tips that made this figure more successful in the revision as well, including grouping things together and text hierarchy. But yeah, so if we go to the next tip, our tip number 2 is about text hierarchy. So now that you have your composition, we really want to focus on key elements. And for a lot of protocol figures, one of the key elements is going to be your text. So just take a few seconds and read what's on the screen right now. It's a little experiment for everyone. Okay, so you would have likely noticed that the text size and the formatting were the main things that really influence the reading order rather than the placement. I fell for this the first time I saw it. You tend to read the biggest, most bolded thing first, and then you're going to go down to that second line. That's the second biggest, maybe that italic. And then a lot of people will miss the very first thing that's at the top because it's the smallest font. So text size and formatting are really often overlooked, but they're essential tools in conveying the intended reading order and information hierarchy. So we can see an example here of a figure that was submitted to us for a previous makeover. And if we go to the next slide, we actually counted a total of 6 different text sizes, in addition to formatting choices like bolding and italics that didn't really seem to convey any additional information that was useful. The figure also generally suffered from a little bit of over design. So there were a lot of kind of unnecessary boxes within boxes. And you definitely kind of want to avoid having that Russian nesting doll situation with too many rectangular design elements because it adds a lot of confusion. So as much as possible, you want to make sure that all of your design elements are very purposeful and strategic rather than just kind of design for the sake of design. So in the next slide over, you can see this is our makeover. We've simplified the composition, of course, and we've removed some of those unnecessary boxes that were cluttering the figure. We also simplified the use of color, which we'll touch on in a later tip. But really revising the text size and formatting is what made the biggest impact for this particular example. So in the next slide over, you can see now that we've reduced the number of distinct text sizes to just 4 instead of 6, and use text hierarchy to our advantage to emphasize the correct reading order. So it's important to use text size and formatting really strategically, which is really to say that the number of different sizes and formats of text should correspond exactly to the number of distinct levels of information. So example, your title, your subtitle, supporting text, footnotes, etc. So every single level of information should have its own formatting, but no more and no less. And that's really how to use effective text hierarchy. So next, we'll talk about grouping elements together. So simply grouping things alone can really make a figure a lot easier to consume. And we do this by consolidating similar or related items together visually and making sure that we put white and negative space around those groups. So when I was still in a lab back in my grad days, I always like to keep my bench very organized. I can't say that about all of my colleagues, but I really wanted to make sure that everything is organized. And the way that I did that was to group things that I knew I was going to use together in the same spot. I would maybe even use a tray to make sure that everything is grouped together appropriately so that I know where all of my stuff is. And it was a lot easier to find what I was looking for when I had a bench that looked more like the example on the right rather than the example on the left. Even though it's the same number of items in both pictures, you can see that it's just a lot easier to find what you're looking for when things are grouped together. So here we have an actual example of a protocol. And in the example on the left, there is really no, there's a few issues here that we can talk about. There's no clear grouping of bacterial cells related to each step, which is really what this figure is trying to highlight. There's also insufficient kind of negative space between each of the steps. They overlap slightly, or they're a little bit tightly close together so that things aren't very distinct from each other in terms of the steps. You can also see that the step titles are basically decoupled from the text description. So we have some steps where the step title or number is at the top, whereas the description's on the bottom of the figure, and then it kind of reverses, and it's a little bit confusing to get through, and it's not really clear how I should be reading the figure. So on the right, we've fixed a lot of these issues by using a couple of grouping strategies. So first, you'll notice that we used call-out boxes, which are the ones that Sally's highlighting there. So those call-out circles or call-out boxes really help visually group the bacterial clusters that are associated with each other together and associate them better with each step in a more distinct way. We've also grouped the related text of each step to the top, and that reduces the cognitive load. So now we have the steps clearly associated with the text that describes them, followed by the actual visual that we're trying to look at as a representation. We've also done something a little bit subtle here at the bottom where we've added redundant legends at the bottom of each step to help keep the steps a little bit more separated. So rather than having to go back and forth between the legend and what each specific bacteria is represented by, it's a lot more seamless this way by just grouping the specific legend that's needed to each step. It just reduces that cognitive load again and makes it a lot easier to follow. Our tip number 4 is to really consider proportions. So poor proportions can make your protocol really difficult to interpret. And this goes on both sides. So you can have a figure where all the elements are too large, it creates a lot of crowding and your eye doesn't really know where to focus. But you can also, on the flip side, have a figure where all the elements are a little bit too small, too sparse, and the key elements that you're trying to highlight don't end up standing out at all. So for optimal clarity, it's really important to strike that balance between emphasizing the key visual elements of your figure while retaining sufficient padding around the elements to avoid that crowding effect. Now, an extra little tip here. So if we go on to the next slide, and I'll actually have Sally help me out with demonstrating this. An extra tip here is, so let's say you have a case where you're trying to show something that by nature has a lot of small detailed components, and you're scared that the key features of your figure is going to get lost and it's not going to be obvious. Well, one option is to kind of just blow everything up and make everything really large. But like we said, that can create a lot of crowding. A better alternative in this case is to actually use a zoom out call or a call out box. And we actually have those in our library. Like you can see, Sally's looking up some options here. So we have different formats and different styles of these. But we can create this to really highlight the key features that we're trying to point out without crowding the overall figure. So you can see Sally demonstrating that. And all we're going to really need to do here is just copy over that main figure. Actually, maybe we'll group all of that together first so that it's all one image. We'll copy it over, and then all we have to do is just blow up the size to the correct zoom level that we want. There we go. Perfect. And then we can actually, so that seems like a pretty good size for the details that we want, and then we can crop it. So you can see Sally here kind of cropping the portion of the well plate that we want to highlight. And actually a pro tip here, if Sally can actually just go back into that cropping option, you can actually choose a crop stroke. So you can create a crop stroke around the cropped area that you want. So maybe we'll make that size 2 or so. And that actually automatically creates a box around the specific area that you're cropping. Perfect. So we probably don't even need that box in the callout, and we can just use that stroke on the crop itself to highlight the area that we're interested in. And then from this point on, whatever details that you wanted to highlight, let's say you wanted to put an antibody in there because this assay involves antibodies, you can just search up an antibody, plop it in, and that way you avoid having to have really, really tiny antibodies in that original form of the figure. Awesome. Okay, great. So that's just a little bit of an extra tip there. And the final tip that we're going to talk about is to simplify. And we've broken this down a little bit into a couple of different kind of sub-tips. So first, we'll talk a little bit about lines. Different types or sizes of lines should have very, very specific purposes and be used in a strategic manner. So you can see in the examples here on your screen that in the original version of this figure, we have a lot of different types of lines, but we also have a lot of different formats of lines, different colors, thicknesses, and that can create a lot of confusion. And actually your eye ends up being drawn to the lines rather than what the lines are trying to communicate in terms of information flow. So we want to be strategic about how we use lines and arrows. So for example, we can use thicker arrows to convey an increase in production of something, or we can use faded arrows to convey movement. A lot of times as a science designers, we'll use dotted arrows to convey a signal that might be weaker or uncertain. So all of these different types of arrows have very specific meanings, and you want to be kind of conservative about how you use them. rather than just using a lot of different colors and shapes and sizes. So you can see that in the makeover there, it's a lot more effective because the errors are all quite meaningful, but they kind of sit in the background and end up highlighting the key features of the actual figure that you're trying to point out. So they're in a supporting role rather than being the main figure here. We also want to consider simplifying shapes and icons. So using simpler representations when possible really helps avoid unnecessary complexity. And what I mean by unnecessary complexity is we can always try to default to a simpler representation and then go up in complexity only as necessary to convey important details. So you can see in the examples that I've shown here, it's often really tempting to have, like a 3D structure of a G protein coupled receptor, for example, in this case, because they look really cool and they're really visually stunning. And we have all of these options in BioRender. You can actually pull these different icons from our library. You can also use our PDB plugin to create these more complex 3D constructions of the protein. So we have all those options, but you want to consider the purpose behind each one and what you're trying to convey. Most of the time, we find that the more simpler kind of default representations of proteins or membranes or nuclei in this case are sufficient to convey what we're trying to show. And we might only want to increase in complexity when we're trying to highlight something, for example, about the phospholipid bilayer that's really important to show, then we go up in that complexity. And finally, in terms of simplifying, we want to really consider color. So minimizing the use of color will really help the figure look a lot more harmonious. So you can see in the examples here in the original, we had about 15 distinct colors. The color palette was not at all harmonious, and it makes it very difficult to draw attention to any key elements in this case. Whereas in the makeover version of this, we reduced the colors down to just 5 distinct colors. The color palette overall is also a lot more harmonious, just kind of sticking to different shades or hues of purple and blue to make it all kind of cohere together a little bit more. And what this really allows us to do is that if we have something about the figure that we're trying to really highlight, we can then use color strategically to highlight those key elements like that MHC receptor that you see there. And that kind of concludes our tips portion. So really, really excited to jump into a live figure makeover at this point. I think it'll be really interesting for a lot of folks here.
Mm-hmm. Okay, so let me transition over from this slide, to over here.
Awesome.
Cool. Do we want to turn our cameras on? Oh, hello. Mina, I think you're muted.
There we go.
Okay,
I'm good now.
Awesome. All right. Yeah, so we're hoping this figure makeover portion will be a little bit more collaborative. So yeah, that's why we turned our cameras on. And by being more collaborative, we're hoping to show off some features we have on BioRender so that we can collaborate in real time. So I believe Mina is also he also has this figure open, and so he'll be popping in, and you might see him move some stuff around. Okay. That being said, let's move on to the figure that we're going to make over. So here is a figure that actually was a figure submitted by one of our community members. So who shared this kindly so that we can make it even more better than it already is. We'll say at the start that it is already quite successful in that a lot of the elements are nicely aligned to one another, and it's really easy to follow what's going on because of the numbers and the arrows. But there are some things that I think could be improved. And I'm wondering, maybe we can open it up to the chat a little bit to see what everyone thinks. Based on our tips and tricks, maybe if you think there's things that could be changed about this figure, feel free to type it up and share with us. Yep.
Right, Linda, yeah, absolutely.
Yeah, there we go. Yeah, so hopefully we're hoping everyone would notice that. So right now, if you follow this figure, you can see that, the top row, it reads from left to right. but then the bottom row, it doesn't, right? It goes right to left, which is sort of counterintuitive to how we generally read things. So easy fix, just select everything. And what you can do is go to the top toolbar and go to the flip and flip horizontally. And that will magically just shift everything so you don't have to do finicky work of, switching all the placements of stuff. Let's see. Okay. Any other comments? See, people are saying things get crowded, need titles.
Yeah, color harmony for sure.
Visual, color harmony. Yeah, this
A lot of really great observations.
Yeah, text and numbers. Yeah, okay. So definitely all of these make sense to me. So for sure. I think let's start with the first one of making the text be right next to the number. So right now you can see that there's these numbered steps, and then there is the text label below, which means the person who is viewing this has to jump from number one to all the text below, which is not ideal. You could group them together. So you know what, let's show off the collaboration function. Mina, do you want to adjust the bottom while I adjust the top?
Yeah, let's do that.
Cool. So you can select all of this, then move it to the top.
So we're both on the same file right now, and we can work on it simultaneously.
And then shift everything a little bit down to make up for the space.
Yeah, I'll do that on my end as well.
So you can see, yeah, the red boxes is essentially Mina doing half of the work for me. Go. Like that. Also, you'll notice that the styling of the text is quite bold, right? So the little numbered circles, they have dark backgrounds. As well, the text is bolded, which you don't really need to bold everything, because if everything's bolded, nothing is bolded, right? So let's go about unbolding. Again, Mina, do you want to do the bottom?
Yeah. And if you're working on this independently and not sharing a video right now, or maybe working at this at different times, sometimes it's really useful to also just if you're collaborating on a file, to share some comments, maybe leave some feedback. So I just left Sally some feedback there about these recent changes.
Nice.
Looks much better. And then Sally can respond. She can also close out those comments. Lots of really great ways of collaborating.
If you'd like. There we go. And then, I mean, there's nothing to resolve here, but I'll resolve it. And so you can know that that comment has been seen and has been addressed. Okay. Cool. I'm just shifting everything down. Hold on. There we go. Let's see. Is there anything else people have mentioned? I think I'm just reading through. Is there anything else that you noticed, Mina, while I go try to?
So we have a question here about how do we initiate the collaboration function? Vaguely recall there's a function where you can leave comments for other collaborators. So I think we just demonstrated that. And you can actually tag people specifically if they have a BioRender profile or account. You can tag them specifically with their name, and they'll get a notification that a message has been left for them on the template that they're collaborating on. Let's see. Some suggestions still rolling in about maybe changing the size of the icons a bit, make them more aligned.
Yeah, for sure. Yeah, so you'll notice that right now there is areas where things seem a bit crowded because there are a lot of things that are maybe a bit bigger than they should be and are close together. So you can easily fix that by just making things a little bit smaller. And just go about doing that throughout the figure. And then once you've made everything a bit smaller, it means you do have more space to sort of to use to add on any necessary components that you need to really make your figure breathe. Just like that. Actually, at this point, this will take a bit of time so for the sake of time, we could jump to one of the prepared slide options we have. Hold on just a second.
Yeah, and we have some still like really astute observations coming in here. So the image feels a bit too clustered. This is from Alexander. The image feels a bit too clustered. Maybe the simplification and union of steps that kind of correlate with each other a little bit more. So yeah, we can definitely do a lot about declustering in this particular case.
Yeah. So if you don't mind, I basically jumped to one where we had that pre-made a little bit. So
yeah.
If we go back to where we were here, the icons were bigger and they're more clustered together. So by making things smaller and then spacing things out, you'll also notice that the callouts, we remove the tails and just put the little circles right next to the test tubes. That often is a stand-in for a zoom out. People generally know that if you do that, that is acting like a zoom in. And to make everything more consistent too, we've added callouts here as well, sort of showing what's in the tube, just to make things a little bit more clear. Okay, just reading through the chat at the moment to see if there's anything else. I think there was a comment about color, yes? Too much color?
And actually, Sally, maybe before we do that, maybe we can very quickly show how to also share. I think a lot of people have questions about
Gotcha.
sharing and collaborating. So we can very briefly kind of go through how to share something that's that you want to collaborate with people on.
Gotcha. Okay, there's multiple ways. So the easiest way is go to share. And you'll see that people at Party have access to this file. If you know the person's email that they have the BioRender account on, you can just feel free to type that email in and then send the invite. I prefer if you just copy the link and then send it out. So to make sure that works. So you want to make sure that the sharing setting is appropriate. So if you're just sharing this for the person to view, feel free to just make it view. But if you're sending this to a person so that they can edit along with you and add comments and things like that, make sure that the setting is at can edit. If it's like that, and if you copy the link, if you now then send that link to someone else, they'll have full access, full editability.
Awesome.
Cool. Right. Getting back to the makeover portion, I think the comment was that there was a lot of colors going on. So definitely agreed with that. Also, we'll note that there is this bright green going on, which, I mean, it's a nice color, but maybe it doesn't need to be bright green. I don't think this necessarily needs to be highlighted. Also, we'll say for people who have red, green color blindness, if you have like orangey color on top of green, next to green color, it can actually not read with proper contrast. So just to avoid that error altogether, you could just go in and change it to another color, like red, for example. Changing it all to red for consistency here. Oh. Oh, sorry about that. Hold on. I was just zooming out there. And also, there are things that could be like, again, so this doesn't need to be green as well. So just let's make everything sort of in a warm tone.
And as you're doing that, Sally, we actually have a couple of questions that are, I think, really important to highlight here as well about aligning things and distributing things evenly. So a lot of people notice that now our numbers are all really well aligned. Things are spaced out a lot more evenly. Are there any quick ways of doing that in BioRender?
Yeah, for sure. So to demo that, maybe I'll just go off the canvas for a little bit and basically drop a bunch of shapes for you that are not aligned and not distributed well. So like that. So you can see that this could have better distribution. So to do that very easily, you could select all the things you want to distribute, go to the top. If you have multiple items selected, you'll see this pop up on the top bar for you. And you go to Align, and you go to this Distribute Evenly portion, and then click whichever way you want to distribute. In this case, I want to distribute it horizontally, so I'll just click that. And you can see that now everything has even spacing. Let's say if you want everything to be aligned, so you can see that all these guys are at different heights. If you want them to be at the same height, you can, depending on what the things are shaped like, click multiple options, but here the top does its job and now everything's aligned.
Awesome. Yeah, so you can align things by the top, by the bottom, by the middle. You can distribute things, and it's really, really easy. Really, all of this is one-click functionality. And that's actually a really important tip as well for protocols in particular, where you have a lot of steps and you want to be really cognizant of your spacing and how everything reads, and making sure that you have really good even distribution and good kind of negative white space around all of your steps and elements. So really important feature that you can use there.
So let me just do it quickly. Let me make this misaligned. So let's say you had it all over the place like this. Let me select it all, distribute, and then bring it all to the bottom like this. Yeah, quick and easy. Cool. Just with the looking at the time here, just few final touches on this makeover. Yeah, so it's sitting at a pretty good spot right now. We've kind of made all the colors warm. We've evenly distributed things, unbolded things, so that the story is very clear. Some additional things that could be done. For example, these icons, the little poison icon, the tube icon, the little cow. It's nice, but it kind of takes away from the figure and it could make it look a bit cluttered. So don't be afraid to sort of remove those. Oh, hold on just a second. Remove those things and just replace it with simple text if needed. Like that. And instead just say plus PFA. I think this is what's happening here. Yeah, and it's much more than streamlined, rather than having to include unnecessary icons to sort of potentially clutter up your image. Of course, make sure the text is not too big. There we go. Yeah, and so we've mocked up the final result for you here for the sake of time. So here's something that we might land on as the final version. And just to show you what the start and the end looks like, there you go.
Amazing.
Yeah, so the same story told essentially, but just a little bit more refined. It was successful before in various ways, but made it even more successful, if you will. Cool.
Awesome. And yeah, we won't, I don't think, have time for a second makeover. We had one kind of on the back burner just in case we had a little bit of extra time, but maybe we'll just kind of skip to our last slide here and just mention a couple of things that might be.
Let's avoid that. Hold on.
Oh. Yeah, so just kind of before we sign off, if anyone has any questions about how to use specific features or different license types, you can always visit help.biorender.com. Has lots of great step-by-step how-tos that are covered in our help docs. You can also contact us directly at support@biorender.com. We will be sending a link out for the recording of this webinar, so be on the lookout for that. And yeah, so we're also launching. No.
There we go.
Yeah, so perfect. Thank you. Yeah, so we're launching a poll right now that you'll see on your screen just before everyone kind of takes off. Just let us know, how we did today on this session. Just any feedback is always really, really appreciated to help us make sure that these webinars are as useful as possible to folks in the future. So for more information on institutional pricing, you can visit biorender.com/contact and complete the form there. Do keep an eye out for an email with the recording of today's webinar, as well as an email from someone on the BioRender team who will be reaching out in case you'd like to request personalized demos for your team and learn more about institutional licensing options. That you can let them know about that as well. The personalized demos are always really cool because we can do figure makeovers with specific figures that you actually bring in, and then you get to keep the final result as well for your personal use, which is always really great. So yeah, thank you again, everyone, for joining today's session, and we hope to see you all in future webinars. Happy illustrating.